stop me, تمام. Okay, so what we're doing here is basically trying to separate cells according to their functional duties. Okay, استنى هي أرجع. Okay, to the functional duties. So now we are trying to look at cells that, let's say, that can phagocytose, that can kill cells on their own, by their own, that can even activate things like cells that can activate T cells, B cells. Yes. So we are just trying to separate cells according to how they function. Okay. طيب, now there are three main functions that cells can lie within, and so first one is phagocytosis. Okay. Second one is cytotoxic cell functioning, and activation, proliferation, and effector activity. So those three things. So we are gonna first look at the ability of a cell to actually phagocytose, okay, and test if these cells actually phagocytose or not. Second one is the ability of a cell to actually go kill, like cytotoxic T cells. And also proliferation, activation, effector activity. So this is basically T and B cells. So you'll see what I mean now when we go through it, okay? طيب, أول إشي, what is phagocytosis and how it's performed, okay? When a bacteria, first step, a bacteria enters the body, تمام? The first thing that happens is that something detects it, such as, let's say, macrophage or whatever. Yes, these or neutrophils. Yes, these are the first linings that actually act without anything. They're innate. So just go in there and try to phagocytose it and stuff like that, and cause more cells to start phagocytosing. Okay. طيب, when something detects it, okay, whether like the first line cells detects it, the first thing that they would do is actually release stuff such as cytokines, okay? And cytokines are proteins that are released by those cells, immune cells, first line from the innate immunity, to cause other cells to come to the spot where that bacteria is present. So this is called chemotaxis, okay? Here I just put down a note saying active movement of cells towards chemoattractants, and chemoattractants are chemokines, for example, or cytokines. So first step of phagocytosis is chemotaxis. So migration, they move towards the place where the bacteria is. The second thing is phagocytosis. And so the whole process of phagocytosis starts with recognition. So, immune cell meets, okay, I have this here. Immune cell meets bacteria. Tamam? So when they meet, they shake hands. How do they shake hands? By so many ways. So those immune cells, they would shake hands with the bacteria using many receptors that can bind to things present on the bacterium. First thing here, if you see, FCR receptor. FC receptor. So what's FC receptor? It basically binds to antibodies that opsonized the bacteria. So generally, in your blood, body just produces antibodies, and these antibodies may be specific or non-specific, and depends on where these antibodies came from, okay? But let's say just an antibody binds onto the bacteria and opsonizes it, okay? This makes it easier for the immune cell to bind to that bacterium, okay? And so why does it say FC receptor? Is because an antibody, okay, usually binds with its FAB region to the bacterium and leaves its FC region exposed. And so the FC region of the antibody that is exposed binds to the FC receptor found on the immune cell. That's the first kind of handshaking, okay? طيب. And just to let you know that the antibody in this case is IgG, okay? طيب. The second way that an immune cell can bind is using a complement receptor, okay? Complement receptor binds to complement proteins, and complement proteins are just tags produced by the body that can bind to foreign substances or bodies, such as a bacterium. So complement receptor binds to the complement protein found on the bacterium, the tag, okay? And finally, they can bind to carbohydrates, and so hence maltose receptor or mannose receptor. Tamam? For those kind of things, this is how this are just general examples of how bacterium and immune cells shake hands. طيب, the second thing that happens, the immune cell swallows the bacteria into a phagosome. طيب, هاي الphagosome will then fuse with a lysosome. Okay? And so when they fuse, the phagosome has the bacterium, the lysosome has lysozymes, ROS, whatever enzymes that is needed to actually break down that bacteria. So when they fuse, now the bacteria and the enzymes or whatever substance used to break down the bacteria is found in one place. So breaking down of that bacteria starts. Now when the bacteria is fully broken down, this phagosome is lysed, and the fractures of those bacteria that were produced will be used to be presented on an MHC2, okay, found on the immune cells, that will be used for antigen presenting to activate more cells. These more cells that are activated will release more cytokines, and so hence more cells will come to that place, and so hence inflammation when there's an infection to that place. Tamam? طيب, so this is phagocytosis. Any questions about chemotaxis and phagocytosis? Clear?
Yes.
طيب, ask me.
Could you repeat the part about? So you said, sorry, I got it. Anyways, could you repeat the part about like the phagosome? So what happens now that we've engulfed the bacteria? What happens in the phagosome?
So when a phagosome enters, it has the bacteria, yes? Now
Okay.
it binds together and fuses with a lysosome, and a lysosome has all of those enzymes in it, yes? So it breaks down the bacteria. Once it's broken down into fragments. Now the phagosome lyses, releases those fragments. Some of those fragments are used to be presented on an MHC2 for antigen presenting purposes. Clear?
Got that part. But what's this FA11 CD68?
I really don't know, but I think
Do you want me to look into it and then tell you?
yes, please. Just حط هيك note and then just tell us on the group, okay? طيب, let's continue. And so now we have MHC2, and so now this is used to antigen present, to activate more cells, okay? طيب, the next thing that they wrote here is the intracellular killing. This is just what I just said inside. It's the binding between phagosome and lysosome contains ROS species or lysozymes or enzymes within the lysosome. And so oxygen dependent or oxygen independent just states to the substances used to break down that bacteria within the phagosome, okay? It can be oxygen dependent by ROS species, or it can be oxygen independent, such as proteolytic enzymes, for an example, okay? طيب, phagocytosis, easy. طيب, now we know what phagocytosis is and how it happens. Now we're going to detect whether our cells are functioning, the phagocytic cells are functioning in case of an infection. So we have a couple of tests. طيب, so when do we use phagocytic cell function assessment? When we suspect an immunodeficiency, our cells are not functioning well, or in case of infections, okay? We will assess this function in case where there is normal white blood cell count, but they are not functioning well, or there's an abnormal white blood cell count, okay? So abnormal count is basically a kind of immunodeficiency. And a normal count, maybe in case of an infection that actually affects those cells and they're not functioning well, okay? طيب, usually what we are mainly looking at is because they are in very large numbers, is neutrophils. Other examples are macrophages and monocytes, okay? طيب, here, some impaired phagocytosis exist because of several reasons. Physiologically, in neonates, infants, in elderly, pathological, in cases where there's primary and secondary immunodeficiencies. This is where our phagocytosis is not actually functioning. طيب, increased phagocytosis capacity in acute infections. It says that in acute infections, we'll have a larger amount of phagocytosis happening. So this is just the whole explanation of what's happening here. تمام? Moving on. طيب, I just explained this. Chemotaxis. Just remember, before phagocytosis happens, things have to move to certain places. And to move to certain places, they need things to attract them. Yes? Imagine yourself going up a mountain, climbing the mountain using a rope. So you're pulling on that rope to go up to the top. Yes? So cells basically are pulling on to things such as complement activation, such as C3A or C5A, bacterial endotoxins, fibrinogen path, IL-1, IL-8. These are all examples of the rope you're pulling on, or the immune cells are pulling on to reach to the site of infection where they can act. طيب. Remember, chemotaxis is basically movement of the bacterial replication, and then the way they squeeze into the cell membrane to reach to the place where they can act. Okay, and here, hence, the chemotactic mediators are whatever we spoke about here that help those cells to reach to the place and start phagocytosis and the immune function. Okay, just explaining what chemotaxis is, just explaining what phagocytosis is. Okay, طيب. طيب, now we explained chemotaxis. Yes, first step. We have tests to help to tell us if cells are healthy and they're able to move. Yes? So, two tests. Using an agarose plate, tamam. Region number, so there's three regions, 1, 2, and 3. Number 2 is where we place our white blood cells. Number 1 is where we keep our chemotactic factor. It's a chemoattractant. Basically, these things. Or a bacteria, okay? Here, we place a control fluid that does not attract the cells, just so in case we just give a place for control, okay? Always, there's in any experiment, a control. Now we place the cells, and so cells start to migrate. If they migrate, amazing. Voila, we got those cells to actually move to the place that we want them to. And so they can actually do chemotaxis. And so this movement is basically horizontal. So cells are moving horizontally towards the chemotactic factor. طيب. And so now we measure the chemotactic index. It's basically an index that tells us the ability or the amount of chemotaxis that those cells can do, the ability of them to migrate. And there's a range, an acceptable range, in which they are able to do the chemotactic function in a good way or a bad way. It just basically says distance A over distance B, distance A over distance B. And then it gives you the chemotactic index. طيب, the second way is using a Boyden chamber. Tell me.
Yeah, so I looked into FA11 and basically what they do is like they're kind of like just general proteins on
Bro,
the surface.
المشكلة مش قادر أسمعك. يعني I can't hear you.
Is it too much noise?
Your voice is very bad. يعني المايك.
Hold on, let me try something. Okay, how about now?
Yes, it's better. Can you state your question again?
No. So basically, I was just saying I looked into the FA11 and CD68, and basically what they are, they're just like proteins on the surface of macrophages, and what they do is they do a lot of things. Like they activate the adaptive immune response. So once they recognize the pathogen, the bacteria, or whatever, okay, they activate the immune response. They release cytokines, and they also act like as a chemotactic, like we were saying. So basically,
Ah,
they're just there to do like what we need to do after we recognize the pathogen in the method. That's
okay.
it.
Okay, I see, I
So
see.
basically, we're gonna see them later, anyways, in recognizing macrophages.
Okay, I
That's
see, I
that's
see.
it.
طيب.
It's not that deep at all.
ماشي.
Like I think we were overthinking it. That's all.
Okay, okay. طيب. Next is the Boyden chamber. The movement, same thing. The movement of the cells here is vertically, and the only difference between the above method and the below method is that there is a filter, and this filter is narrower, has pores that are narrower than the diameter of the cells, hence to detect the ability of the cells to actually pass and squeeze through a very tiny pore. Tamam? And also has a chemotactic factor on the bottom, right here, and we place the cells here, and so they cross. This filter right here has pores which are narrower than the cells themselves, and so if they move through, cells can migrate, okay? And I don't know, for some reason you incubate it in 37 degrees for 3 hours. طيب, next. So now we tested if the cells can move, okay? 2 ways, agarose plate and Boyden chamber. طيب, next. طيب, now we're gonna detect it's a phago test, basically. What a phago test says is that, okay, now the immune cells have migrated. The next thing they will do is phagocytose, or absorb bacteria in them, right? And so we have tests that help us figure out if our cells are functioning well or not. طيب. A phago test is basically when we label our bacteria with fluorescent dye or fluorescent substance, and we measure either how many neutrophils phagocytosed bacterias, okay, or we measure how many bacteria were phagocytosed by one cell. So there's 2 different ways just to tell us whether phagocytosis happens. طيب. Two types of cells here that are analyzed. They're separated according to their size and forward scattering and granulation. So size and granulation, and they just give us according to where they are on the size and granulation of their structure. And this circle right here tells us granulocytes, and we can see that they are fluorescent. Hence, analyzing them, we can see that they have actually phagocytosed the bacteria. In this case, we are analyzing only monocytes, and they are also fluorescent because they have bacteria in them. And so hence we know that they actually phagocytosed the bacteria, okay? So this is a phago test. So first thing, we analyzed using the agarose gel and the Boyden chamber movement. The second way is we analyzed phagocytosis by labeling, fluorescently labeling those bacteria, and detecting them using what? So of course, this is a flow cytometry. It's a flow cytometry graph, histogram or graph, that is drawn after cytometry, flow cytometry being performed on those cells containing those fluorescent bacteria, okay? And so it gives you those results. Just going back to what we took earlier. Okay, I'm gonna move on, tamam? Okay, now, when they're inside the white blood cells, they're not just gonna chill, right? They're gonna be broken down. There are even tests for those, okay? طيب, before I go into those tests, what are ROS species or reactive oxygen species? They are present within lysozymes, right? Lysosomes, right? We said phagosome containing bacteria, lysosomes containing ROS bind, and then they break down this bacteria. طيب, we can test if this reactive oxygen species are functional inside those white blood cells or not by using some tests. طيب, before going into those, what are some reactive oxygen species? We have the superoxide anion, hydrogen peroxide, hydroxyl radicals, singlet oxygen, hypochlorous acid, nitrous oxide, okay? So all of those can actually do the reactive oxygen species function of breaking bacteria down. طيب. Some also phagosomal enzymes present that can also I don't know what those are. Maybe those are the things that actually produce ROS. I have no idea. If you guys can correct me. But so we know what ROS. Yeah.
Yeah, you're right. So
Okay.
that's basically the things just like catalyze the formation of the ROS because you don't want the ROS all the time. So once they're activated, once the cell activated, and then these enzymes are activated, they catalyze the reaction to produce ROS, which then attacks the.
Okay.
You don't want it full time. You know?
Yes, yes. So exactly. So because why don't we need those ROS to be active all the time is because they are actually toxic and they can actually interfere with our let's say mitochondrial metabolism to actually produce ATP. So in this case, they have to be always inactive. To activate them, these are the enzymes that are responsible to activate those ROS species, like oxidase NADPH complex and myeloperoxidase MPO. Now we have ROS species. How can we use them to break down bacteria? طيب, this is just telling us how they are produced. Okay, so H2O2 or O2 is produced from the ETC, and then they're getting activated by those enzymes to actually kill the bacteria. Or they can be inside the lysozymes and activated to actually then kill the bacteria. Okay, so two places. And this is just telling us that impaired oxygen bursts, sometimes like we have more than needed ROSs, is because in case of diseases, or increased oxygen bursts in infants or young children, or in case of severe infections. Tamam? But this is ROS. Okay, now we're going to see how ROS is analyzed, if it's functional or not. We go do a phago burst test. طيب, what's a phago burst test? The whole general idea about a phago burst test is that ROS generation results in the chemical oxidation and its fluorescence. They will put a chemical, and this chemical will reach the place where ROS is functioning. Then ROS oxidizes this chemical. This chemical changes color or gives out light, okay? Hence, tells us that ROS are actually functional, okay? This is just telling you that neutrophils are better in phagocytosis and oxygen killing than monocytes because it has a percentage of oxygen burst capacity more than monocytes. طيب? We have 10 minutes to go. I'm gonna cut it after 10 minutes and restart it. طيب. So now we first finished chemotaxis, the agarose gel and Boyden chamber. We then moved into phagocytosis and we did a phago test. Okay. Phago test where we just put fluorescent bacteria and measured how many bacteria inside the white blood cells. We then now are going to actually dive into how if those bacteria are broken down inside the phagocyte, inside the phagosome, okay? And so we use chemicals. The first chemical, there's 3 tests, but the most important one they actually pointed at is this one, nitro-blue tetrazolium NBT test. طيب. We will incubate this NBT with the cells that the white blood cells, yes. And so let's say we incubate as well bacteria in there. Bacteria swallows the immune cells swallows the bacteria and then takes in the NBT as well. Okay. Now it starts to wanting to break down that bacteria. Yes. So the phagosome will have the ROS and the bacteria in it. The ROS oxidizes NBT, turns from yellow to blue crystals, formazines. And hence you'll see it under the microscope like this. So those cells are functioning. Those cells are actually breaking down the bacteria using ROS. Okay? Easy? طيب. The next thing is a second test to test oxygen burst is chemiluminescence test, I think, yes. In response to stimulus, phagocytic cells emit light. Its intensity reflects the cell's ability to produce ROS. So in this case, color change. In this case, light production. and the third one, cytochrome C reduction test. So those three tell us oxygen burst is functioning. Tamam? Again, three things we did: chemotaxis, agarose, and Boyden. Just swallowing test, basically labeling the bacteria with immunofluorescence. Third is the oxygen burst test using change of color, such as NBT, or fluorescence, such as the chemiluminescence test, or the cytochrome C reduction test. طيب? Easy. طيب, now we're going to do a different function, test a different function. Let's go back here. One, easy, yes? Testing chemotaxis, testing phagocytosis, and then testing ROS functioning in this one right here. And we discussed the tests. Now going to number 2, cytotoxic cell functioning. And so the cells that actually can function as that, such as cytotoxic T cells, natural killer, NKT, gamma delta T cells, all of those are able to kill cells on their own. So we have lab tests to actually tell us if these specific cells are functioning and doing their function. Okay, so we go here. طيب. How is cytotoxicity performed in the body? Recognition, they shake hands. Activation, proliferation, so they become active. Yalla, I want to kill the cells. And the effector phase, which is made up of two ways. Either they kill the cells by toxic granules and death receptors, and both result in apoptosis. The only difference is that toxic granules use perforins or granzymes. So how can those two ways of effector phase lead to apoptosis is by activating a cascade inside the cell called caspase. Caspase cascade. طيب. Perforins and granzymes, they can either activate the caspase directly or indirectly. Directly is by cleaving the caspase. Indirectly is by actually activating mitochondrial way to activate the caspase and then cause apoptosis. Death receptor, on the other hand, activates apoptosis directly by activating the caspase directly. They don't do it indirectly. Okay, just very deep things that we may not need, but I just wanted to point out in case you had questions. So here, effector phase, toxic granules, pores, what is called, like sacs, they just bind to the cell, releasing perforins and granzymes inside to activate caspase to do apoptosis. The second one, death receptor, is called FAS. Remember, we took FAS in class. So FAS is the death receptor, where cytotoxic T cells use the FAS and FAS-L. Like FAS-L is FAS ligand. Those two bind and so activate apoptosis. Tamam? So this is cytotoxicity. So how can we test this? How can we know if those cells are cytotoxic? First one, using radioactive substances. طيب, now clear everything you have taken before. Now we're talking about different function. Okay, عندك you have a bacteria and you have a cytotoxic T cell. Let's say, for example. طيب, the first thing we have to do, get a test tube, put in the bacteria, put in the radioactive substance. The radioactive substance goes inside the bacteria. Okay? Now, we expose the bacteria containing the radioactive substance, such as chromium in this case, to cytotoxic T cell. So cytotoxic T cell will start acting and kill the bacteria, yes? Break it down. We measure before this happens, there's no radioactivity, zero. طيب. After the bacteria is broken down, there is radioactivity. Hence, cytotoxicity is good. طيب. How does radioactivity appear after cytotoxicity happens? Is because the cytotoxic T cell breaks down the bacteria now has pores, its structure is broken down, yes? So the radioactive substance would be released into the solution. And so the solution becomes radioactive. So it's that simple. So target cell is the bacteria, swallows the chromium, The chromium is inside the target cell. Effector cell is a cytotoxic T cell. Those two react. This is broken down. This is released. Now centrifugation happens, whatever it is. And so now we are able to detect the radioactivity. So radioactivity increases means more released chromium, increased cytotoxicity of effector cells. So cytotoxicity and radioactivity in this case are proportional. Both increase. If radioactivity increase, then we know cytotoxicity is increased and is functional. So that's the first way, using radioactive chromium.
Yo, I've got a question, bro.
Hmm?
You say the when radioactivity increases,
Yeah.
that's because the membrane is being ruptured and the substance is being released, right?
Yes.
But surely it would like be radioactive anyways, even if it's inside the cell. So how would you know?
Maybe chromium is not a strongly radioactive material.
I think it is, but like actually, okay, you know what? I'll look into it. Thank you. Sorry, bro.
Because like it says detected by a Y counter, for example. Yeah, but actually, that's a good question. Just send all of those things into the group, but that's just a general idea.
Okay.
Okay. طيب. Number 2. طيب, we have to just concentrate here. The second way to actually detect cytotoxicity is using an MTT test. طيب. An MTT test is basically saying if the MTT stays, okay, if the MTT stays, then the cytotoxicity is high, I think. Wait. So MT is reduced to formans in metabolically active cells. Yes. So if the cells stay alive, so we have let's say, let's go back to simple stuff. We have a bacteria. This bacteria is incubated with MTT. Okay. And we put cytotoxic T cells with it. Yes. Okay. Now, if cytotoxic T cells don't kill the bacteria, MTT, it leaves it alone. So it's actively functional. If it's actively functional, it switches. طيب, let's continue. If okay, so MTT test is the second test that tests for the cytotoxicity of the cell. Okay, now we get a test tube. We put the bacteria inside this test tube. 1 second. Okay. طيب, we get a test tube. We put this, we get a test tube. We put this bacteria inside of this test tube, and then we put something called MTT. طيب, MTT is now absorbed by the cells, bacterial cells. Bacterial cells are usually metabolically active, yes? They're metabolically active. They will convert MTT into formazine. Okay, so keep that in mind. Now, if I put in cytotoxic T cells, they will kill the bacteria. طيب, they will kill the bacteria. The bacteria stops becoming metabolically active. Stops becoming metabolically active. Now, if they're metabolically inactive, they will not convert MTT into formazine. So the solution will stay yellow. Okay? So when we see yellow, or we see MTT is still present, this tells us that cytotoxicity is active. If we see a purple solution, this tells us that cytotoxicity is not active. Did you get this? Did you get this?
Yes, so basically you put the target cells and the effector cells, the cytotoxic cells, and MTT. Okay, so what would happen is the effector cells would activate target cells to make them be inactive. Therefore, they cannot produce formazan. They cannot produce MTT to make formazan. So therefore, it stays MTT. It does not go to formazan because the effector cells act on bacteria to make it inactive. Therefore, you have MTT. But if it didn't activate and bacteria is active, it will reduce MTT to make formazan, which is perfect. So the higher the number you have in the spectral photometer, the closer to purple wavelength, I mean, the more metabolically active bacteria, the less cytotoxicity, and
Yes,
vice versa.
exactly. وانا ما سمعتش منك الا اخر sentence بس yes, that conclusion is good. If formazan is present, then we know cytotoxicity is not okay. If MTT is present or yellow, if it's yellow, we know cytotoxicity is present. Okay, it's confusing here, but no, just know if it's blue, cytotoxicity is not okay. If it's yellow, cytotoxicity is okay. طيب. طيب, now this is just telling us that flow cytometry is able to be used to detect what is it called? To detect cytotoxicity. Why? Is because all of those tests that maybe we have spoken about earlier can be detected using shining of fluorescence. Okay, so for example, I mentioned Caspase activation. Okay, Caspase activation. Caspase activation is what we talked about here. Using effector cells, right? They do their function by toxic granules and death receptors. Both of those things activate caspase to cause apoptosis, right? And so flow cytometry is the third way, third way to detect either even more specific cell killing, because it can differentiate between whether it's apoptosis, cells are dying because of apoptosis, or cells are dying because of necrosis. And so I think what they do is actually label some certain molecules or secondary molecules within those cascades inside the cell, and so they can detect whether the cell is going through apoptosis or necrosis. Hence, they can gate for different molecules, as I see here, annexin V or propedium iodide. So those things maybe are different things within those cascades that can tell us whether the cell is getting broken down by necrosis or apoptosis. This is in case of the target cell. Or we can also label perforins, granzymes, or death ligands and detect the function of an effector cell using flow cytometry. Of course, viable means live, and so those cells do not activate those cascades inside of them, and so flow cytometry does not detect any of those labeled secondary molecules inside that cell. So it tells us there's no apoptosis nor necrosis happening inside, and so you get this region of viable alive cells. Okay. تمام. Cytotoxicity. طيب, now we are done with cytotoxicity. So how many did we do? We did one. One was, again, going back, chemotaxis by agarose gel or Boyden chamber. Two, we actually test whether the cells are swallowing or not by labeling the bacteria with fluorescence. Three, we test the ROS active function by using NBT, chemoluminescence, or what that was, or cytochrome C, right? And then we have cytotoxic cell function. In this case, we use chromium as a radioactive substance, or we use flow cytometry, or we use the MTT test, the confusing MTT test with formazan, okay? Done. So now we're going to the third step, detecting the function of the cells, whether they are able to activate, proliferate, or act as effector cells. And those three regions, basically focusing on T cell and B cell. طيب. This is just telling us how B cells get activated. They go into clonal expansions, increasing their number, they proliferate. So here we are going to look at the activation. We're going to look at the proliferation, okay, and differentiation. And then we're going to look at the effector function of how they actually attack the bacteria. And each and every section of this life cycle includes tests that test for the function of that specific stage of the life cycle of T cells and B cells. طيب. Activation, how they shake hands, right? We have tests for that. 2, proliferation. So how they increase in numbers to make an army that can attack. 3, how they actually cause an effect on and how they kill, basically, or activate, or whatever it is. Okay? طيب. Now, activation is the first thing. I don't really know so much about this, but mitogens and antigens are basically fragments that help activate those T cells and B cells. Mitogens, the basic meaning of this word is basically saying that it is able to activate non-specifically. So it binds to anything and it can activate anything. Antigens, on the other hand, they're specifically bound to specific things that we need them to bind to, such as, let's say, a B cell that has a specific antibody. And so we know what antigen can bind to that antibody on that surface. And so that antigen can activate that B cell. Mitogens, on the other hand, can activate using many things. And so agents used to activate T cells, such as those ones listed here. I don't know if we need to memorize them, but just memorize maybe, for example, the ones that you actually are familiar with, like maybe staph tox, which is toxin A, B, E, right? These are enterotoxins used to actually activate the T cells. And these are different ways in which we can activate T cells, either direct TCR cross-linking, indirect TCR cross-linking. Maybe memorize one for each. And so if they're asking you or about any way of mode of action, you know a material used to activate the T cell. طيب. So this is how we expose and activate the cells, mitogens and antigens. Non-specific activation is mitogens. Specific activation is antigens, okay? First step done. Now we go to the next step. It is the proliferation. What is the meaning of proliferation? It just means increase in number to create an army. And so if we create an army, we need to know how, we need to know if this army is actually created, if this one cell is actually going to create an army, right? So what we use, again, we go back to radiation. In this case, we use tritium labeled thymidine. What is thymidine? It's basically the T. Thymidine is incorporated into the DNA. The more thymidine labeled tritium labeled thymidine present in that solution just means that more DNA is present within more cells, right? So this tells us that there are more cells in the solution. What do? Label the thymidine in the DNA with a radioactive substance called tritium. We label them, and then we allow them to proliferate. And then we go and detect that radioactive, measure that radioactivity. And so more the radioactivity, more the proliferation. That's tritium labeled, okay? Second thing, Burdu. Bro, how about you send me a text with your question? Because wallahi, I can't hear you.
Can you hear me now?
We like all I hear is like people around you. كتير ناس.
But you can't hear me.
I can hear you, but it's very low.
Okay, bro, forget about it.
Hello?
Yo, okay, bro, okay, it's fine. I'll just look into it, bro. Thank you.
اكتب لي question تبعك هون إذا بعرفه I can answer it. طيب؟
It's like a bit irrelevant, but it doesn't. I don't think like I should waste everyone's time by asking it because it doesn't help anybody else.
والله مش سامعك مان. اوكي بعت لي برو، والله مش سامعك. مش عن جد انت قاعد بمكان اظن مليان دوشه.
Okay.
يلا ماشي. Okay, so Burdu. Okay, so Burdu is we actually also incorporate it into DNA. طيب. And then once Purdue goes deeply inside the cell, we denature and expose the DNA and Purdue complex. طيب. Now we add a primary antibody to the Purdue DNA complex. This primary antibody just binds to Purdue. That's all its function. And then we add a secondary antibody, but it's labeled with a fluorescent body. And so this secondary antibody with the fluorescent body binds to the primary antibody. So now what do we have? We have DNA, Purdue, primary antibody, secondary antibody labeled with fluorescence. And then we take it to test with microscopy or imaging, an imaging technique. Okay. And so we then see or test for the presence of this Purdue within DNA. And so we're just talking about proliferation here again, yes, but we're just labeling with a different material called Purdue. And because it's not a radioactive substance, we actually use a fluorescent substance, but we use that antibody, primary antibody, secondary antibody sandwich system. DNA, Purdue, primary, secondary with fluorescence, and then we detect that fluorescence with either flow cytometry or two other methods, such as colorimetric, yes, and chemiluminescence. And so colorimetric is basically an enzyme that digests substance to change color. So that secondary antibody can either be bound to a fluorescent substance to release light, or it can be bound to a substrate. When exposed to an enzyme, it digests it and changes color. So 3 different ways: flow cytometry using fluorescence, colorimetric, change of color because of the digestion of that substrate bound to that secondary antibody in the sandwich, or chemiluminescence, change of release of some form of light when that substance bound to the secondary antibody is digested or oxidized, releases light. So these are just methods to detect that antibody label that is bound to that Purdue, that is bound to that DNA. And so the more DNA, the more fluorescence, the more light, the more results on flow cytometry, right? And so we use flow cytometry to actually gate for different things, like you know, I know how flow cytometry works now. So it just separates them into different things. Again, so lymphocyte proliferation assessment. The first one was using radioactivity, and the second one using fluorescence of light, changes of color because of that substrate digesting the enzyme on the secondary antibody, or because light is produced because of an oxidized agent. Tamam, that gives us, you know, more DNA, more cells, so hence more proliferation. طيب. The third way is CFSE dilution assay. CFSE is basically an ester, and this ester binds also to DNA, and this ester becomes more fluorescent when it's broken down by esterase. So if we are in the first cell, okay, we have CFSE here, it's bound to DNA. Once it proliferates into 2 cells, okay, it divides, yes? So the DNA splits into 2. Hence, we know that CFSE becomes less with the generations. Because as DNA splits, the number of ester that was originally in the first cell decreases, right? So hence, when we see this, now we start at a large number of CFSEs in that one cell, and then it proliferates, yes? Now, CFSE decreases in number, right? But we know that overall, the number of cells are increasing. So less CFSE with time means that there is more proliferation or DNA present. People may ask, why at this certain amount of CFSE, there is a large number of cells compared to this number of CFSEs, because maybe at this stage cells stop proliferating, some of them stop proliferating, others continue, right? Yes, so the number of CFSE measured at that stage of those cells is a lot. And then some of those cells proliferate, causes a decrease in the CSFE overall. Okay. So again, flow cytometry. طيب. These tests again. طيب. عندك activation. We have mitogens, antigens. Second thing, we are testing for the formation of an army of cells. How? By DNA. Testing how much DNA is present. How? By labeling that DNA with first radioactive substance, by labeling it with Burdu, by labeling it with CFSE, and that's it. طيب, effector. Effector is basically, it's saying release of cytokines. What's the cause? What's the use of this cytokine? To attract more cells. To attract more cells to do what? Phagocytose, kill, whatever. Yes. And so we have different types of tests used to detect cytokines. Like, what are cytokines? They are proteins. Proteins used to do what? Chemotaxis, attract more cells. Now we have tests or lab diagnostic methods that will help us detect the amount of cytokines released from those cells. Yes? First, we have intracellular staining of cytokines. We just basically stain the cytokines inside the cells. Because they're proteins, we are able to actually stain them using antibodies. So basically what we do is actually lyse the cell a little bit, put antibodies that are specific to those little cytokines. The antibodies would go deep inside the cell, right? And so bind to that cytokine. So hence intracellular detection of cytokines. And then expose those cells that have labeled cytokines to flow cytometry. And then we can detect the different cytokines that are produced by different cells, helps put those cells and know what cells are present. يعني مثلا, you have T helper 1 can produce IFN gamma. What we did here, we basically lysed the TH1 and then put in the solution antibodies that are specific for IFN gamma. And so antibodies would go inside TH1, bind to IFN gamma, put it in flow cytometry, detect that this cell is TH1. Okay? Same for all others. That's the first method. Second method, ELISA pot. Okay, ELISA. What is ELISA? And what is ELISA pot? And what is Western blot? These are all the three left that can detect what? Cytokines. First, we said intracellular labeling. Basically, we label them inside the cells and using antibodies, and then expose them to flow cytometry. ELISA in general. ELISA is just an arrangement of an antibody sandwich. ELISA pot, same thing, uses the same system. What's the difference between all of them? ELISA, let's go through ELISA first. ELISA and ELISA pot. ELISA basically detects the presence of cytokines and the concentration in a snapshot. ELISA pot detects the increase of the concentration of cytokines across time. So the only difference between those two is the time factor. يعني أنا هسه حطيت كاسة, now I put a cup that has cells that release cytokines, and I detect how much cytokines is present in this solution. That's it. Now, ELISA pot, I don't have cytokines, but I expose cells to ELISA pot and I allow them to release cytokines. And with time, I detect how much cytokines is present compared to when first it started and when it ended. That's the only difference. طيب, how do we do this? What's ELISA in general? I know it says ELISA pot here, but they just use the same antibody system, but the only difference between them is the factor of time. Okay? طيب, ELISA. ELISA is basically, we have a pot, we use a capture antibody. That's the first thing we do. A capture antibody, from its name, it captures cytokines. But what's the state of it? It's immobilized. We put it on the dish, it's immobilized, leaving its fab heads upwards. Now we put cells that release cytokines. once the cytokines are released, the cytokines that are specific to be able to be bound to that capture antibody bind to it. So we want the cytokines that we want to detect. Yes, in case of Th1, we want IFN gamma. Okay, so IFN gamma, or those cytokines, bind to that capture antibody on the top. So what do we have right now? Petri dish, capture antibodies, IFN gamma, or cytokines bound. طيب, what's the next step? The next step is we have a primary antibody. A primary antibody binds to that cytokine. Okay, so now we'll have capture antibody, cytokine, primary antibody, or detection antibody. Yes? طيب, what next? We add a secondary antibody. A secondary antibody is bound to a detection method. Let's say a fluorescence or a substrate, okay, or an enzyme. Yes? And so this secondary antibody binds to the primary antibody. The next thing we do is we add the substrate. The substrate is digested by the enzymes found on the secondary antibody. Once they're digested, they change color or release light, and hence gives us the ability to detect where or how much there is cytokines in our solution. طيب, let's repeat. Let's go to a better way. طيب, here, yes, just to give you a visual way. Sandwich ELISA. How ELISA works? You have أول إشي Petri dish, or whatever this dish is, yes? Now we add capture antibodies, and it's just not one, it's a lot of them on all of those circles. Capture antibody is placed, it's immobilized. The next thing we add is a target protein. What is a target protein in our case? We want to detect effector, right? So in this case, we have cytokines. Cytokines that are specific to that antibody. Our interest cytokines bind to our capture antibody, okay? The next thing we add is a detection antibody, or a primary antibody, okay? The primary antibody binds to that specific cytokine. The next thing we do, we add a secondary antibody that contains or has on its surface an enzyme. And in this case, the enzyme is called HRP, horseradish peroxidase. Okay, so horseradish peroxidase. طيب. Now, this sandwich is created. طيب, there's no change of color. How can we detect where the cytokines are? We add a substrate, in this case, TMB. When TMB is added, this enzyme would digest this substrate, changing color. The change of color tells us exactly where the cytokines are, and we can calculate how many are there in there. And so we can detect whether this cell actually produces cytokines and effectively does its effector function, releasing of cytokine. طيب, this is in a snapshot. That's an ELISA. طيب, an ELISA pot is different. We just do the same exact thing, but we use time. How much cytokines are released per time, okay? طيب, last thing, Western blot, okay? Western blot is basically used for the same reason, okay? But in this case, the only advantage that Western blot can give us is that it gives you the presence of the cytokine plus the size of this cytokine. And that's the only difference between ELISA and Western blot. And hence, because of the size, it can actually differentiate it by electrophoresis. Okay? And that's it. That's our lecture. طيب. Any questions? Also, quickly, I'm just gonna quickly go through it. Quickly. First thing. Phagocytosis by phagocytosis by three main things. أول إشي, you have yes, chemotaxis, اللي هي the movement of the agar plate. Yes, here, phagocytosis by agarose plate and Boyden chamber. Then we are testing the absorption by a phago test, labeling the bacteria by fluorescence. Then we are testing for the oxygen burst by using nitroblue tetrazolium NBT that switches into formazine, change of color, chemiluminescence, and cytochrome C reduction test. Then we go into the second function, cytotoxic cell function. We have radioactivity, chromium, or we have MTT, the confusing test, or we have flow cytometry. Then we go into the effector function. So activation proliferation effector, right? Activation by using mitogens and antigens. Proliferation is by using DNA. We label DNA here. Tritium, Burdu, CFSE. Third way is effector. We actually test the cytokine released. And so here we do the first method, intracellular staining of cytokines. Second method, ELISA and ELISA pot. Third is Western blot. And that's it. This is the practical that we did. Done. Okay, do you guys have any questions?